Thanks to Dan and thanks to Michigan Sea Grant for inviting me to talk about the Cooperative Science and Monitoring Initiative. I'm going to focus more on kind of lower trophic levels. So essentially, a really sort of close look at things that are at the base of the food web. So we're going to talk about benthic invertebrates and phytoplankton more zooplankton. But essentially, the things that the fish are eating. And we're also going to talk about a very particularly important part of a fish life, and that's the larval phase. So just after they hatch. And so I'm just going to jump right into it here. I'm going to start with just a real brief introduction, a little bit about myself, and then talk a little bit about the process of CSMI, because I think it'll be really good for you guys to sort of hear about, this is essentially like how managers get information about the lake. Then I'm going to focus very much so on the benthic community assessment, talking a lot about quagga mussels and what's going on with quagga mussels and things on the bottom of the lake. And then talk later about zooplankton and larval fish surveys that happened during the last CSMI cycle. So first of all, just a quick introduction. I am, for better or for worse, I'm very much a product of the Great Lakes region. So I am a research professor at Purdue University. I work in the Department of Forestry and Natural Resources. I am one of about 5 or 6 aquatic faculty, people who study aquatics or fisheries in the Natural Resources Department. I also work with Illinois-Indiana Sea Grant. So right there on the border with you guys. I'm the Great Lakes Ecosystem Specialist for Illinois-Indiana Sea Grant. So I try to take more of like a holistic view of the ecosystem in the Great Lakes. And I run applied research programs and bring in graduate students to Purdue University to study essentially lower trophic levels. A lot of the stuff that I've done is supported by the EPA, and here in Chicago, there's a group called the Great Lakes National Program Office, and this is the group of from the EPA that is responsible for monitoring the offshore conditions of the Great Lakes. And so you may have seen this ship out on the lakes. This is the Lake Guardian. It's the largest research vessel that operates in the Great Lakes, and pretty much operates. It's going to be going out here in a couple weeks, and it pretty much operates during the entire open water season and covers all 5 of the Great Lakes. So I've been working in the Great Lakes now for about 20 years. I started off, I grew up in Ohio, and then took a brief stint down south and went to Samford University in Alabama for my bachelor's degree. But then I came back up to Southern Illinois for my master's degree, where I worked on fisheries in reservoirs. But since the early 2000s, when I started my PhD program at Ohio State, working in Lake Erie, I've been working in the Great Lakes. So I did my PhD at Ohio State, and then I also did a postdoc stint at University of Michigan, and then right to Purdue. So I've been in and around the Great Lakes for quite a while. So one of the things that I do in my position is, as Dan mentioned, is I help to administer the CSMI, or the Cooperative Science and Monitoring Initiative. And so if folks haven't heard of this program, CSMI it's a binational effort. So it's the United States and Canada. This program is written into the law of the Great Lakes Water Quality Agreement. So this is something that the United States and Canada are obligated by law to participate in this program. Hold on, let's shut something here. Okay, thanks. So it is the goal of CSMI is to provide enhanced science and monitoring activities that are conducted in response to the priorities that are established by the lake partnerships. And so I have the lake partnerships here in bold because these are essentially the people who manage the lakes. So these are people like your Michigan DNR representatives. There are also, tribal biologists who serve on the lamps. There are people who are in Eagle, the Great Lake, the more of the water quality groups. But these groups are essentially looking for information to help manage the lakes. And that's what CSMI is offering. So it's to provide science and monitoring information to make management decisions on each lake. So it's done on a rotating basis. So it's an every 5-year cycle because we have 5 lakes. There's an emphasis on one lake each year. So that's essentially how it works. This is how the process works. Just a different view of it. But again, I want to emphasize the Lake Partnership is the group of managers that they take input from people like yourselves. They listen to their constituents, and they hear about what are the issues on the lake and what are the things that we need, the science that we need to manage the lake. And then once every 5 years, they come up with this document that is the science priorities for their lake. So this is just a list of these are the things that we need to know to better manage Lake Michigan. And then that goes to a group of federal agency people, the CSMI task team. And then they work to essentially design research projects that can be done in 1 year to try to answer those questions or those science priorities. Not everything can be answered using, this sort of like one 1 year research project, 1 year assessment survey. But they try to identify the ones that can. And then that goes into years 2, 3, and 4. And then I also want to point out this year 5. So it's also important that we sort of close this loop. So we look at what the results were and communicate the results. So that's part of, what I'm doing here today is trying to take the results from CSMI and make sure that everyone hears about, what happened and what sort of information we got to address those science priorities. So there's places here where, here in year 5 and when we're reporting out the results, where the people who are interested in the lake, the different user groups, you should be hearing about the science that happened. And then I also wanted to emphasize there's the part where there's the developing the priorities. So you don't be shy about, making sure that the managers hear the concerns that you have about the lake, because there's actually a mechanism for them to try to get information about that. So specific to Lake Michigan, the previous cycle, this is the cycle that we worked on. So the previous cycle's priorities were set in 2018. I'm just wanted to point out here that around 2020, just like everything else, COVID screwed up this cycle too. So there's a little bit of sort of overlap, the federal research ships weren't really allowed to go out in 2019 and 2020. So things that sort of get messed up here. And there's the results that I'm going to talk about are field work that happened in 2020 and in 2021. But essentially we are in this sort of reporting out, and we're actually to the point where we're setting new priorities for the next cycle. So in 2025, there's going to be another CSMI on Lake Michigan where people are going to be out, a ton of different researchers are going to be out collecting data based on the priorities that have been set in 2023. So I just wanted to real quick, just kind of take the pulse of the room here. I wanted to see, so if you guys are thinking about the things that are affecting Lake Michigan, I can't really see the group in the room, but I just wanted to sort of have this little straw poll to see, according to this group, what are the most pressing issues affecting Lake Michigan. And so you can feel free, if you're on virtual, put a number in the chat, 1 through 4. If there's a way to this is not going to be quantitative at all. And I just wanted to see what the general ideas were. Or if there's something else entirely in those first three that I'm not thinking about, you could feel free to throw something else in there. And I'm going to give you about like 20 seconds, 30 seconds or so.
In a 2, a 1, 2, 3, 2, 3.
Fewer threes than I was expecting. All right, we're getting a lot of threes online.
I think that's everyone. 2s and 3s.
All right. Yeah. So, just so if you're sort of thinking about these things, in my view, a lot of these things are sort of interrelated too, and I'm sure in your views as well. But again, like different people who are using the lake for different reasons. Someone might really be concerned about like water quality in the lakes as well. So you got to sort of take into account all the different opinions and the different ways that people use the lake as you're setting up these priorities. But back in 2018, there was this official sort of priority setting process that happened, and these were basically the 4 priorities that popped out. So the things that I'm going to talk about today, which again are the results of like kind of the 2021 CSMI effort, they were trying to address these 4 priorities, and so and they're very broad. So addressing nutrient food web dynamics in a changing ecosystem, a lot of this has to do with things that are happening because of quagga mussels. Another one that like I don't think about very often, but probably very naively, the concerns about contaminants and bacteria. The bacteria I know especially came up about like beach closure issues in Lake Michigan. There was a priority that was centered around addressing nutrients that are coming in from the watershed and tributaries and how that it's affecting Lake Michigan water quality. And then I thought this was pretty interesting. The number 4, the managers wanted help in trying to connect to the stakeholders. So in a lot of this introduction that Michigan Sea Grant did, they talked a lot about social science and connecting, with stakeholders and what do the stakeholders want. That's something that the managers hear. And so this is in one of their priorities for the 2020 CSMI. So in terms of what actually happened, though, again, not everything is really amenable to this process. But these were the specific targeted projects that happened in 2024. There was a broad food web sort of study looking at trophic levels and small-bodied fish. There's intensive sampling along a transect off of Muskegon, Michigan, where NOAA has a field station. Probably of interest to this group, there was a lot of work that was done around lake whitefish, tracking lake whitefish movements in Green Bay and also in northern Lake Michigan, more near Traverse City or Grand Traverse Bay. There's some thought that maybe we're undersampling very near shore areas. And so there was actually a program where people went out and did beach seining to look at fish communities in very, very near shore areas, which is really less sampled. Another thing was this really spatially comprehensive larval fish study, and then monitoring contaminants in the ecosystem. For today's presentation, I'm going to talk about these 2 that are in bold. So this broad food web study and this spatially comprehensive larval fish survey. If these other things sound interesting to you, and I hope they do, we actually have a full report that's available. So all of these different projects are described in detail in this report. And if you hit this QR code, you should be able to, it should take you to a place to download that as a PDF. And I'm not going to stay here too long, but it'll be on the last slide as well, this QR code. But okay, so now I wanted to sort of jump into one of the things that happens in CSMI pretty much every year is this lake-wide benthic community survey. This is essentially like almost all of the information that we get about the spread of quagga mussels in the Great Lakes comes from this program. So in 2021 in Lake Michigan, there was a very large research survey that happened in over, you can see July and September, and there were 99 stations that were sampled For benthos, or all benthos, 114 total, and there were also 19 other stations where they were looking just for Dreissena and Dipraia, and we'll talk about those. But this map is showing where all of these stations were, so it's a pretty extensive spatial coverage. In order to pull this off, again, you need a lot of cooperators. So it was EPA and NOAA, and there's a group of benthic ecologists from Buffalo State that were aboard the Lake Guardian. Again, this is that EPA research vessel. And the methods that they use, so they're essentially going out and using what's called a ponar dredge. So it's a big, heavy scoop that sort of goes down and takes a chunk out of the bottom. You bring it up and you look at what's there. They're also using benthic imaging cameras to sort of look at, actually get a visual representation of what's down there on the bottom as well. And then, so basically you're just doing that at all 119 of these stations. And then this is the type of data that you get back. So this is going back, to the 90s. This is basically showing the spread of quagga mussels in Lake Michigan. So back in 1994, 95, there were no quagga mussels in Lake Michigan. The concern at that point was zebra mussels were starting to establish in the Great Lakes. Quagga mussels are closely related, but a different species. But by 2000, we started to see the establishment of quagga mussels in Lake Michigan. And then here, as you see, as you go on, those like darker colors essentially represent higher densities. So lighter blues are lower densities. The blacks are where you're seeing the highest densities of quagga mussels. So as you can see, essentially the lake is sort of filling. We're getting quagga mussels kind of everywhere. And the densities, they may or may not really be going up. They certainly are changing in different areas of the lake. But in 2021, we found quagga mussels at 98% of the sites that were sampled. Quagga mussels represent 75% of invertebrate density, so the numbers of invertebrates, and 99.7% of all of the biomass in the benthic community is quagga mussel in Lake Michigan, which is not available too much for fish production. So this is biomass that's not really being used all that much. There's also like oligochaetes, so small worms were the most diverse and most common benthic organism after Dreissena. Is about 20% of the density. And the communities differed by like different depths rather than different spatial areas of the lake. So you have a different community that's in the deep water relative to near shore, and that's pretty consistent wherever you are in the lake. One thing that's very concerning is that Dipraia is declining in the lake. Dipraia is, if you're not aware, it's a benthic invertebrate that at one point was one of the most important pieces of the food web, a very important prey item for a lot of the deeper water species. Actually, kind of everything ate it. But Dipraia was a really common invertebrate. Here's a picture of one. Just a little, like some people call them scuds, that were on the bottom. But as you can see, in the 90s, before quagga mussels were established, they were sort of all over the lake. And as it's essentially the opposite of what you're seeing with the Dreissenid mussels. As Dreissenid mussels take over the lake, the Dipraia are increasingly found only in kind of the deepest areas of the lake. And so this is something that has, really important food web implications, because we're taking this benthic biomass that used to be in Dipraia, used to be available to promote fish production, is now going into quagga mussels, where for the most part it's a dead end. And then, so just breaking things down by depth, and so these graphs are showing this first one down here, this whole lake. This is essentially the biomass of Dreissenid mussels, mostly all pretty much quagga mussels through time. This is just so you can see kind of the trend, and we have an increase, but it's not accelerating. If anything, it seems to be plateauing out a bit. And over here, further on the right, this is the different densities or the biomass of Dreissenid mussels by different depth. And so the important take home here is that in shallow and in their deepest sites, so in less than 30 meters, less than 90 feet of water, we're seeing an increase in Dreissenid mussels there at these mid-depth regions. The at least the biomass is sort of plateaued off, but where we continue to see increases is in the deepest part of the lakes. So greater than 90 meters or about 270 feet. The other thing that came out of this survey, there is a new invasive invertebrate in the benthos. The New Zealand mud snail is expanding. We're finding that in 3 times the sites that they were found in 2015 in the 2021 survey. And they seem to be replacing the native snails. So about 93% of the snail density in Lake Michigan is this newly and non-native. It's yet to be seen if it's going to be really detrimental or become invasive. But it's a non-native species that's coming into Lake Michigan. So to summarize, quagga mussels dominate the Lake Michigan benthic community. This is one of those pictures from the benthic imaging system, and this is what the bottom of Lake Michigan looks like in a lot of places. A lot of mussels. Over the past 10 years, the quagga mussel population is kind of stabilizing, but there's definitely is increasing biomass in the deepest zones of the lake. Unfortunately, Dipraia continues to decline. It's only present at very low abundances, and we're only seeing them in the deepest parts of the lake. And then, video analysis might be a way that we can assess this more rapidly in the future. In terms of management implications, Dreissenid mussels, they dominate the benthic community, and they have widespread ecosystem impacts. So all that mussel biomass is filtering out a lot of the phytoplankton that would be sort of driving the food web and making its way up to fish. But for the most part, it's just all it's doing is staying on the bottom as Dreissenid biomass. Dipraia, which is a really high quality food item. These are really fatty little invertebrates. They continue to be low, and fish are shifting their diets, out of necessity. So the Dreissenid mussel trajectory, though, is relatively stabilizing. Again, we're kind of leveling it off. So hopefully this could mean that the impacts of the Dreissenid invasion are going to stabilize as well. We hope maybe we won't see quite as much of the biomass of the phytoplankton taken out as the Dreissenid population stabilizes. So with that, I'm going to stop for this section and just see if there are any questions.
There is a question in the chat wondering about the relationship between quagga mussels and what fishermen refer to as green slime.
I'm not 100% sure what green slime is. Does anyone have a...
Cladophora.
Oh, okay. So yes. So it's thought that Cladophora is benefiting from the Dreissenids. So Dreissenids, again, like they're taking all the nutrients and then they're just sort of recirculating it at the bottom, and so like there's less nutrient, less algal production that's happening within the water column, and it's shifting all down to the benthos, which is where this like Cladophora. So they're getting nutrients from these mussel beds. And yeah, probably they are, for the most part, helping to sort of fertilize the Cladophora a bit. Let's see. Are they filtering out contaminants in the lake? For the most part, yes, but a lot of contaminants, they do tend to bioaccumulate. And so, meaning that like if a fish eats another fish, it's going to take on that contaminant load from the fish that it eats, and it's going to accumulate that. And so a lot of the contaminants that are in the Great Lakes historically have been these kind of bioaccumulating contaminants. And so for the most part, you see higher, burdens in the things that are higher up on the trophic levels. In terms of eradicating quagga, I'm not sure that's really going to be possible. I know there are things that people are doing. There are similar to what they did with lamprey, where they developed a poison that was specific to lamprey. People are looking for a poison that's specific to quagga mussels, to dry-scented mussels. That's something that people are doing right now. There's a lot of work that's being done to try to build reefs, like rocky reef structures in the Great Lakes. I'm actually involved with a project in Saginaw Bay where they're getting ready to build some new reefs out in Saginaw Bay. And I know that there's a material that they're testing that seems to be resistant to quagga mussels settling on it. And so at least for when they're constructing reefs, they're considering the material and trying to think about ways to incorporate materials that are resistant to the quaggas. Let's see, any information on mysis? That's a good question. Mysis, there was the comment earlier about coho being in the southern part of the lake, and I know one of the thoughts is they're down there eating mysids. That's a place where there are a lot of mysids, and there is in the report. I'll say that I don't have it off the top of my head, but if you download that report, mysids is a important part of the food web. It's something that is measured. My general understanding is that populations are declining, but it's not as precipitous as what's happening with Diparia. And then, do you see any many midges? We do see so chironomids. We see midges. They are less of an important part of the benthos in offshore. They're more found in nearshore areas. And one thing I do know is the fish definitely find them. They're very common in fish diets. But we see fish are better at finding them than we are. It seems like.
We can take a couple questions here in person too.
I was just curious about what the abundance of spiny water fleas. Seems like around here the alewives are eating them. We have them early in the year and then they disappear. And I was wondering what percentage of the biomass do you think are the spiny water fleas?
Okay, yeah, that's a good question. I am going to talk about zooplankton a little bit in the next section here, but I'll say this: I had a graduate student in the 2015 CSMI, so not this cycle, but the one before, where we actually looked at Bythotrephes or spiny water flea. We looked at their abundances through time, and they are they become a very important part of the zooplankton assemblage. It tends to be, their populations tend to peak in the fall. One of the things that we found in that study, though, was that we actually never saw the population increase through the fall, and then we ran out of ship time. We didn't actually get to keep going to see it go back down. And so they tend to go up in the fall. They become the dominant zooplankton basically during the fall. But we don't know like when that comes back down. And that is actually really important because we don't do all this monitoring late in the season. So we really are probably underestimating how many spiny water flea are out there, because we're not out there sampling when they're at their highest levels. So they are important, and we're probably underestimating how important they are.
I'm gonna ask about, was there any conclusions made on, when you were showing the maps of densities of the Dreisseniid mussels on the southern tip of the lake, it was showing colors of white instead of any blues? Is that just, because of depth, or?
We'll see. I could go back to it. No, I mean, it was just like there are fewer there. I mean, those are real. There's actually data being collected there.
Any conclusions why? This is my question.
Oh, I mean, anything I would say would be just sort of, you know, I would say there's probably, you know, it tends to be much sandier down there, and you know, quagga mussels kind of do well on any substrate, but they actually, you know, sometimes sand and then high wave action, they don't do really well. So maybe that would be my best guess for that.
It says the quaggas are filter feeders. When they eat that plankton, they either bioaccumulate it or they excrete it. So is that fertilizing all that excrement? Does that turn over when the lake turns over, when it warms up, or how is that used by more phytoplankton?
So yeah, when you do get sort of turnover, that those nutrients are they are available throughout the water column. But turnover tends to happen, you know, when water's about 4 degrees. And so it's not during a time when there's high enough temperatures to really like kick in a lot of the algae. There's diatoms that are definitely produced during that time. But most when the water column becomes stratified, it's essentially staying down there. It will, but you are right. Yeah. When you get turnover, when you get an isothermal, like a single temperature water column, those nutrients do become available, but it's not at a time when things are really biologically very active.